Hi, my name is Priscilla Chong, a Bachelor of Agrotechnology student from the Faculty of Resource Science and Technology in UNIMAS. Today, I will be presenting my industrial training experience at the Sarawak Biodiversity Center SBC. SBC is a leading research institution in Sarawak that focuses on biodiversity conservation, biotechnology research, natural product development, and the sustainable utilization of biological resources. Throughout my internship, I was primarily attached to the algae research section under the supervision of Dr. Anne Mary. When I first joined SBC, my expectation was to gain practical laboratory experience and understand how scientific research is conducted in a professional setting. However, as the internship progressed, I realized that the experience extended far beyond laboratory work. It exposed me to the biodiversity conservation, sustainable agrotechnology practices, community engagement programs, public outreach activities, and multidisciplinary research collaborations. Therefore, the objectives of my industrial training were to gain practical exposure to biodiversity research, enhance my technical and professional competency, understand sustainable agrotechnology applications, and develop skills that would prepare me for my future career. The majority of my internship was spent in the algae research section. My daily routine usually began at the algae cultivation facility, also known as ACF, where I collected algae samples from different cultures using sterile syringes. The samples were transferred into centrifuge tubes and brought back to the algae research laboratory for processing and analysis. At first, these activities appeared repetitive. However, I gradually understood that routine monitoring is one of the most important aspects of algae cultivation because even small changes in culture conditions can significantly affect growth performance and productivity. Through this experience, I learned that successful research often depends on consistency, patience, and attention to detail rather than complicated procedures alone. After the sample collection, I perform routine processing and analysis. One of the techniques I frequently used was cell counting using a hemacytometer. The procedure involved loading a small volume of algae culture onto the counting chamber and observing the cells under a microscope. The cell density could then be calculated to determine the growth performance of the culture. Besides cell counting, I also perform microscopic observation to assess cell morphology, contamination, and overall culture health. Through repeated practices, I improve my technical skills, accuracy, and confidence in handling laboratory equipment. More importantly, I learned that scientific data collection requires precision and consistency because reliable data forms the foundation of research decisions. Apart from laboratory analysis, I was also involved in algae cultivation management. My responsibilities included algae initiation, harvesting, media preparation, culture maintenance, acclimatization, and topping up nutrients at the cultivation facility. One important lesson I learned was that different algae strands require different cultivation approaches. For examples, some strands require specific nutrient formulations because the nutritional requirements differ. In addition, certain strands such as AL571 are sensitive to excessive sunlight and high temperatures. To protect these cultures, shade structures are installed. However, while shading reduces environmental stress, it may also reduce light intensity and potentially slow down its growth rates. This taught me that successful cultivation requires balancing multiple environmental factors rather than focusing on a single parameter. During my internship, I was also given opportunities to learn techniques outside my primary section. One memorable experience was learning cereal dilution and agar plate streaking under the guidance of my supervisor, Dr. N. This was my first exposure to these techniques. The procedures required strict aseptic practices to prevent contamination and ensure accurate results. Although I was initially nervous because the techniques were unfamiliar, the experience strengthened my laboratory confidence and broadened my understanding of microbiological procedures. This demonstrated the importance of stepping outside of my comfort zone to acquire new knowledge and skills. My internship also exposed me to several other research sections. For example, at the plant preparation lab or plant preparation station, I assisted in preparing biological materials such as pomelo skins, daun gelam, limau susu, and rumput malaya for further processing and essential oil extraction. In the plant tissue culture laboratory, I learned acclimatization techniques that successfully transferred pokok bakak planters from agar media into peat moss. Moreover, at the Ganoderma Cultivation Facility (GCF), I assisted in waste management activities and learned the importance of maintaining cultivation hygiene and production efficiency. These experiences helped me appreciate the multidisciplinary nature of research at SPC. Now, among all the activities I participated in, the Lawas Community Engagement Program or Traditional Knowledge Trip to Lawas was one of the most meaningful. The program involved visits to Long Musu and Long Karabangan communities. Unlike laboratory activities, this program allowed me to see the practical impact of research on society. I observed how SPC researchers work closely with local communities to promote sustainable utilization of biological resources through initiatives involving pokok bakak and rumput malaya. This experience taught me that scientific research is not only about producing data, but also about creating positive impacts for communities and supporting sustainable development. Another memorable experience was the biodiversity documentation trip to Gunung Gading National Park. This activity allowed me to observe biodiversity directly within a natural ecosystem. The through flora and fauna observations, photography documentation, and biodiversity recording activities, I developed a deeper appreciation for conservation efforts. The trip reminded me that biodiversity research begins with understanding and protecting natural ecosystems. Without conservation, many valuable biological resources may be lost before their potential benefits can be discovered. One of the highlights of my internship was participating in Sarawak Biodiversity Day 2026 here at SBC. The event attracted visitors from various backgrounds, including students, families, researchers, government agencies, and community representatives. I was assigned to the algae research booth, where I explained algae cultures and research activities to visitors. Initially, I was nervous about speaking to the public. However, as the event progressed, I became more confident in communicating scientific information. This experience significantly improved my communication skills and taught me the importance of making science accessible and understandable to society. Throughout the internship, I encountered several challenges. In algae cultivation, different strands required different nutrient formulations and environmental conditions. Some strands were highly sensitive to sunlight and temperature fluctuations, requiring careful monitoring and cultivation management. I also observed operational challenges, such as rust formation on photobioreactor support frames due to prolonged exposure to moisture and outdoor conditions. These experiences taught me that research involves continuous problem solving, adaptation, and improvement. Challenges should not be viewed as obstacles, but as opportunities to learn and develop better solutions. Looking back on my internship journey, I believe the most significant outcome was my personal and professional growth. Technically, I gained competency in algae cultivation, microscopy, cell counting, media preparation, molecular biology techniques, plant acclimatization, and also cultivation management. Most importantly, I became more confident in working independently while maintaining responsibility and accountability for assigned tasks. In conclusion, my industrial training at Sarawak Biodiversity Centre, SBC, has been a highly rewarding and transformative experience. The internship enabled me to apply theoretical knowledge from my agrotechnology studies in a real research environment or setting, while developing practical, technical, competency, and professional skills. Most importantly, it has shown me that science is not only about generating knowledge, but also about creating meaningful impacts for society, the environment, and future generations. Thank you.